Hi everyone, I'm Hongkui Zeng from the An Institute for Brain Science. Here I'll give the Tyler Rock lecture on understanding brain cell diversity. And here are my disclosures. The collective goal of neuroscience is to understand how brain works and how its function goes awry under different diseased conditions. This is a daunting task given the extreme complexity of the brain itself, with millions to billions of neurons and a similar number of non-neuronal cells, as well as trillions of connections among the neurons. To tackle this complexity, it is essential to take a multi-level approach to unravel the mechanisms underlying brain structure and function at all these different levels. At the An Institute, one of our major focuses is to understand the complexity of the brain at the first two basic levels, cell types and connectivity. We have built multiple technology platforms to reveal the diversity of cell types in the mammalian brain, from mouse to human, and how the different cell types are connected to each other to form functionally specific circuits. We presented our large-scale data sets to the community as foundational resources to facilitate the entire field's discovery. Why studying cell types and how? To understand any complex system, such as the brain, an essential first step is to create a parts list of that system. For the brain, the parts list would be a composition of cell types. By obtaining the parts list, we'll also be able to gain genetic access to different cell types, which will then allow reproducible investigation and discovery across the different labs. This effort will allow us to better understand other important biological processes such as development, evolution, brain circuit and function, and eventually uncovering the cellular basis of various devastating brain diseases. To obtain a comprehensive catalog of cell types, it is critical to use a systematic, unbiased, and scalable approach, striving for completeness. We need to make multiple standardized and quantitative measurements and use a data-driven approach to classify cells into types. Importantly, because brain cells exhibit tremendous diversity at multiple levels, from molecular to anatomical to physiological and functional, it is also critical to profile individual cells across these different modalities and seek concordance across modalities in terms of their cellular properties to arrive at a unified definition of cell types. Over the last decade, single-cell technologies have really revolutionized our way of understanding cellular diversity and function in biology. We have taken advantage of these different single-cell technologies and established different pipelines and platforms for characterizing and defining cell types in the brain. At the molecular level, we use single-cell transcriptomics, single-cell epigenomics, and spatially resolved transcriptomics to profile tens of thousands to millions of cells to derive a molecularly defined cell type taxonomy for different brain regions. At anatomical level, we use full morphology reconstruction to obtain the dendritic and axonal morphology, as well as axon projection patterns, and use those to define anatomically defined cell types. Electron microscopy data generation at an increasingly larger scale will also give us the opportunity to obtain not only local morphological, but also synaptic level connections between different individual neurons. That will also enable us to define cell types based on morphological and connectional properties. Those anatomical approaches combined together have been really powerful in defining cell types in other species such as the Drosophila. To bridge molecular information and anatomical information, we can use approaches such as barcoded connectomics, in which individual neurons are labeled by barcoded viruses, and the correspondence between molecular profiles and axon projection patterns can be obtained for single cells by sequencing of the barcoded transcripts. Other methods such as patch-seq will allow us to obtain triple modality information from individual neurons that have been patched in brain slices, and a combination of intrinsic electrophysiological properties, transcriptomes, and local morphologies from the recorded cell allows us to define integrated MET types. And those MET types can serve as critical Rosetta Stones that will then allow us to assign molecular identities to the cells for which only certain type of information is available. For example, only morphological information is available, such as neurons reconstructed from electron microscopy dataset. We also use retro-seq and a related method to again assign molecular identities to connected cells, neurons from different brain regions. And combining in vivo functional imaging, such as calcium imaging, with post-hoc spatial transcriptomics profiling allows us to assign molecular identities to cells for which we have obtained functional properties from. Finally, generation of cell type targeting tools and is a powerful approach that will enable a wide range of investigation of the structure and function of molecularly defined cell types. Traditionally, we have been doing this by generating transgenic driver mouse lines, targeting specific cell types. In recent years, we have been able to use the combination of single-cell RNA-seq and single-nucleus attack-seq data to identify chromatin accessibility sites that are putative enhancers for specific cell types. And we can test those putative enhancers in AAV viral vectors, and in a pretty good success rate, we are able to obtain enhancer viruses that target specific cell types. This approach has very important implications because it will then target cell types not only in traditionally genetic model organisms such as mouse, but also it can be applied to non-model organisms, in particular non-human primate, and even the human brain itself, which then can serve as not only research tools, but also therapeutic means to deliver functional genes to specific cell types in the brain. So overall, over the last few years, there have been many studies from the field in utilizing this whole set of technologies to gain a better understanding about cell types in the brain, about how to define them properly, about the correspondence between different cellular properties from molecular to anatomical to functional, and in particular about testing and validating transcriptomically defined cell types and elucidating their functions. In the interest of time, in this talk, I will not be able to talk about this large body of multi-modal cell typing work, except for one example case, which I will talk about next. Instead, the main focus of my talk will be just on the single-cell transcriptomic approach that we are taking, with the goal of deriving a first iteration of cell type atlas and the general initial framework for cell type organization from individual brain regions to eventually covering the entire brain, that we believe will serve as a foundation to enable not only functional studies of the specific cell types in the mouse, but also to enable cross-species comparison and the understanding of the organization of cell types in not only animals, but the human brain itself as well. So Our starting point is a single brain region. And here is an example of a collaborative effort that took place within the Brain Initiative Cell Census Network, which we are part of, in creating a multi-modal cell atlas for a single cortical region, the primary motor cortex, across several mammalian species: mouse, macaque monkey, marmoset, and human. This diagram shows the integrated cell atlas that we obtained, which was anchored on the mouse transcriptomic taxonomy and with additional cellular information attached, including the correspondence of the transcriptomic cell types or clusters with traditionally defined cortical cell types, both neuronal and non-neuronal, with the relative proportion of the cells for each type, and with morphological and physiological information that we obtained for those cell types. So in this work, not only we generated this multi-modal atlas for primary motor cortex, we have also uncovered several major organizing principles of cell types in cortical areas, and that can be also generalized into other brain regions. And those principles are: first, the cell types are organized in a hierarchical manner with different levels of definitions of cell types, from class to subclass to type, occupying different major or minor branches of this hierarchical tree of cell types. The different modalities of cellular properties can be integrated together to create a unified definition of cell types with different levels of granularities. And the integration involved the integration of transcriptomics, epigenomics, spatial distribution, morphology, projection properties, and electrophysiological properties. We find the coexistence of discrete and continuous variations of cellular properties across this entire cell type taxonomy tree, with discrete distinctions between major classes and subclasses of cells at a higher branch level, whereas continuous variations mainly exist at the terminal leaf or cell type level. There are also individual differential variations among the different modalities of cellular properties, for example, between transcriptomics and morphological projection patterns. And those differential variations also exist mostly at the terminal branch level. Similarly, we observed the coexistence of cross-species conservation and divergence of cell types. Cross-species conservation mainly exists at the major branch level for cell classes and subclasses. In particular, we defined 25 subclasses of neuronal and non-neuronal cells that are highly consistent, that are conserved across the different species. Species-specific divergence, on the other hand, mainly was observed at the terminal leaf or type level, with often fuzzy correspondence between individual clusters derived from different species, as well as a substantial amount of individual gene expression variations. About 20% of the genes would have differential expression patterns in different species, even for the same conserved cell type. So in this next slide, we further illustrate the conservation and divergence of cellular properties across species. On the left-hand side is a consensus transcriptomic taxonomy tree across human, marmoset, and mouse for the primary motor cortex. The major cell types, including all the subclasses and a few major types below the subclasses, are well conserved and aligned across species. At the same time, you can see that there are also a variable number of clusters within each of those major types, and the clusters do not exhibit precise one-to-one correspondence across species. On the right-hand side, we illustrate the differential morpho-electrical properties for a major subclass of neurons in the cortex, in the motor cortex, and that is the layer 5 extratelencephalic projecting, or ET, layer 5 ET neuron types. We can see that the morphological and the electrophysiological features are very significantly different across species. Despite of these differences, all these neurons from the different species can be aligned into a single major neuronal subclass, the layer 5 ET, because they also share major molecular transcriptomic gene expression signatures. So this is an example of a study for cell types from a single brain region. Next, we asked how do cell types vary across different cortical areas, given that different cortical areas perform very different functions. In this mouse study, we profiled 2 different cortical areas at the opposite end of the cortical sheet: primary visual cortex at the back and anterolateral motor cortex in the front. We obtained a similar hierarchical structure of the different cell types. A major finding from this study is the observation of a dichotomy between the 2 major classes of neurons. Namely, the GABAergic inhibitory neuron types are common or shared between the 2 different regions. On the other hand, the glutamatergic excitatory neuron types are highly distinct between the regions. We think this dichotomy may be originated in the development of the cell types, with all the glutamatergic excitatory neuron types generated within the cortical plate itself, and thus were subject to morphogen gradient defined regional difference or regional specificity. The GABAergic inhibitory neurons, on the other hand, were generated in the subcortical ganglionic eminence and migrated into the cortex during development. As such, they may not carry region-specific information in their transcriptomes. Further extending that work into covering the entire cortical sheet, in this next study, in which we profiled more than 30 individual regions in the isocortex and hippocampal formation of the mouse, so that we can have a more comprehensive view of cell type variations across the many different cortical areas. The regions covered include all the sensory, motor, and higher order association areas in the medial, prefrontal, and lateral part of the isocortex, also known as the neocortex, as well as all the regions within hippocampal formation, considered as a more ancient archicortical region, including hippocampus itself, its nearby subicular complex, as well as the lateral and medial entorhinal cortex. And 1.2 million cells were profiled, and the single cell transcriptomes give us a much expanded taxonomy tree shown here, still in a hierarchical organization. The principles we discovered in the 2 cortical area study still apply in this much larger study. And however, the principles have also evolved with additional complexity discovered. Specifically, in the GABAergic class, we find that most of the cell types are still shared across all the cortical and hippocampal regions, as shown by these rainbow-colored bar graphs. In addition to that, we also now identified a set of cell types that are highly specific to hippocampus itself. So the coexistence of shared and region-specific GABAergic types. On the glutamatergic side, the number of clusters has expanded dramatically. There are still a number of clusters or cell types that are specific to particular regions, but at the same time, we also observed that many more cell types are now shared between different regions, especially among the neighboring regions, and there exist all kinds of gradient distributions of those shared cell types in dimensions such as medial, lateral, or anterior, posterior. A major finding from this study is the relatedness of the glutamatergic neuronal types from hippocampal formation and those from the isocortex. First of all, the glutamatergic types from those two major brain structures are highly distinct from each other, as illustrated by this UMAP representation colored by regions or subclasses. All the isocortical neurons are present in those major continents in the UMAP, representing the major subclasses identified from cortex, including the intrathalamus-cephalic or IT neuron, subclasses from all the cortical layers, from layer 2-3 to layer 6, the layer 5 PT or ET, subcortical projecting neuron subclass, and then the deep layer, layer 6 cortical thalamic projecting, the layer 6B subplate neurons, and the layer 5-6 near projecting or MP neurons, which form a distinct continent by themselves. Besides those major continents occupied by cortical neurons, the glutamatergic neurons from hippocampal formation are all present in those many tiny islands near the continents, suggesting that the glutamatergic neuron types from hippocampal formation or HPF are highly distinct, not only from the cortical neuron types, but also from each other. However, in addition to that distinction, we also observe that there's a very specific homologous relationship between HPF glutamatergic clusters and cortical neuronal types or subclasses. By performing a homology search, we are able to find highly significant homologous relationship, one-to-one homologous relationship between HPF clusters and each of the layer-specific cortical subclasses of glutamatergic neurons. And furthermore, this molecular homology is supported by similar laminar distributions of the corresponding pairs of types. And this is supported by the identification of specific marker genes for each of the HPF clusters and their laminar specificity of that indicate where the cell types are located. One interesting thing I wanted to point out is that the layer PT neurons from the cortex, which are the major output cortical subcortical projection neurons from the cortex, has the greatest homology with the hippocampal CA1 and subicular neurons, which don't form this kind of a typical, laminar specificity. And this molecular homology is interestingly correlating the similar long-range projection patterns, because CA1 and subicular neurons are also the major output projection neurons from hippocampal formation to a variety of subcortical regions. So to summarize all the rich information about the glutamatergic types together, when all those types are arranged by their spatial specificity in this graph here, with 240 clusters and 75 supertypes organized by the regions where they come from in columns, isocortex on the left, and hippocampal formation on the right. And they are also organized by their laminar distribution patterns, with the superficial layer intrathalamus-cephalic neurons at the top, deep layer intrathalamus-cephalic or cortical projecting neurons here, the layer 5 extrathalamus-cephalic ET or PT neurons in the middle showing here, and their homologs, corresponding homologs. And finally, the layer 6 cortical thalamic neurons and layer 6B subplate neurons at the bottom. So this organization illustrates a parallel cell type organization between hippocampal formation, and isocortex, with all the major glutamatergic neuronal subclasses in isocortex also identified in hippocampal formation, suggesting that there may be a evolutionary relatedness between those two major brain structures in terms of their cell type composition. And further, we would like to propose that such homologous cell type molecular relationship may also give rise to similar connectional properties for the cell types in those two different structures. And this is a testable hypothesis. Finally, by looking at the overall organization of cell types across the entire cortical sheet, we can see that the combination of region-specific cell types and a gradient distribution of shared cell types collectively define regional specificity for each cortical region. So it's not like a cortical region is defined by a specific region-specific type, but it's the combination of all the different types together, unique and shared together. Furthermore, this transcriptomic landscape also defines different modules in the cortex, as shown in this hierarchical tree of the different cortical areas that was obtained by meta-clustering of the transcriptomic clusters. And this transcriptomically defined cortical modularity is very similar to the cortical modules that have been discovered in numerous previous connectional functional studies of the cortex. And it suggests to us that the transcriptomic cell type defined cortical modularity may be underlying the modularity of connectional and function in the cortex, including that of the human cortex itself. And that's something we would love to be able to test in the future. Okay, now lastly, we have now further expanded our single cell transcriptomic work to cover the entire mouse brain. And here is just a basic summary of all the major brain regions that has been covered, and just explain the terminology a little bit here. In the telencephalon, there's the two major parts. There's the cerebral cortex, or developmentally the pallium structure that covers all the cortical regions, including the isocortex and hippocampal formation I just mentioned in our previous study. And it also covers the olfactory areas, including piriform cortex, which is also considered a more ancient cortical structure, as well as the cortical subplate regions, which include structures such as claustrum and basolateral amygdala complex. In addition to the pallium, the telencephalon also includes the subpallium structure, which is called the cerebral nuclei, that includes striatal and pallidal areas. In addition to the telencephalon, we also have profiled the entire thalamus and hypothalamus, which compose the diencephalon, as well as all the regions in midbrain and hindbrain and cerebellum. This is yet another large-scale collective collaborative effort within the BICCM, and which we now call the BICCM 2.0 Whole Mouse Brain Consortium. In this consortium, several labs have been working together and generating a whole brain scale molecular data sets at a single cell level. And those include single cell RNA-seq, single nucleus RNA-seq, single nucleus methylation-seq, single nucleus attack-seq, as well as 3 different sets of whole brain scale spatial transcriptomics data using the Murphy method or the slide-seq method. We are now in the process of integrating all these different data types together across the consortium to create yet another integrated cell atlas for the entire mouse brain. As that work is ongoing, in today's talk, I will just tell you about our work at the Art Institute in using the combination of two large-scale whole brain data sets to generate a high-resolution cell type atlas for the whole mouse brain. And those two data sets are a single-cell RNA-seq data set with 7 million cells profiled, and also a set of whole MERFISH data set using the Visgen MERScope platform with a 500 gene panel that was designed based on our single-cell RNA-seq taxonomy by choosing the most powerful combination of marker genes. The marker genes with the most discriminating power, discriminating different clusters. So here is the end result. This is the single cell transcriptomic atlas we generated by combining the two data sets together. So to arrive at this, we first QC'd our single cell RNA-seq data set to select a 4 million high quality single cell transcriptome and use those to perform clustering analysis, which results in a taxonomy of 5,200 individual clusters. We then mapped every single segmented cell from our whole brain MERFISH dataset, which also contains more than 4 million individual cells, by mapping each of those MERFISH cells into our 5,200 cluster RNA-seq taxonomy, we are able to assign cluster identities for each of the MERFISH cells. And then in turn, we use those MERFISH cells to for each cluster to assign the spatial or anatomical location for each of our transcriptomic clusters. So by combining those different data sets and results together, we are able to derive a transcriptomic taxonomy that is organized in a hierarchical manner, with those 5,200 clusters grouped into about 1,000 supertypes, 300 subclasses, 32 classes, and 7 divisions. This taxonomy tree shown here only shows the cell type organization at subclass and above level. We're not able to show more details at a cluster or supertype level here. The 7 divisions include 3 non-neuronal divisions, glial cells, vascular cells, and immune cells. And then there are 4 neuronal divisions with all the pallium glutamatergic neuron types forming a distinct branch, and the GABAergic neuronal types from the subpallium forming another distinct branch, and other highly distinct neuronal types from cerebellum, cortex, main olfactory bulb, and other areas from another mixed distinct division. And all the other remaining neuronal types in the middle part of the brain, which are highly divergent, but also with different high degree of similarity with each other, forming a very large neuronal division. The taxonomy or cell type atlas can also be visualized in a UMAP, which contains all the 4 million single cell transcriptomes. It is colored by class, subclass, region, and the major neurotransmitters each cell expresses. A key point I want to mention here is that we find cell types are not just defined by their molecular properties, molecular features, such as which neurotransmitter they express. But the cell types are also strongly influenced by which brain regions they come from. So in the end, the computationally derived cell type organization, for example, those 32 classes, are really defined by the combination of molecular identities and regional identities. Furthermore, we discovered different cell type characteristics among different brain regions. In this graph, we can see that the number of clusters from different major brain structures is vastly different. And the numbers of clusters do not scale proportionally to the number of cells that have been profiled. For example, in the pallium glutamatergic part, even though we profiled orders of magnitude more neurons from all the different pallium structures, the number of clusters is still moderate. And the numbers of clusters in thalamus and cerebellum are relatively low, whereas on the other hand, the numbers of clusters in hypothalamus, midbrain, and hindbrain are very high. By analyzing differentially expressed genes among all the different clusters, or DE genes among all different clusters, we also find that interestingly, there are only a small number of DE genes between each pair of clusters that are from hypothalamus, midbrain, or hindbrain areas, suggesting that the clusters in those regions are all pretty similar to each other, with few DE genes that can differentiate them. The other structures, such as pallium, thalamus, or cerebellum, on the other hand, have a wide range of the numbers of DE genes between each pair of clusters, suggesting that the cell types in those regions are highly divergent. And also, some are similar to each other, whereas others are very different. So a wide range of similarities and differences in those regions. Furthermore, by looking at the 3D spatial span of each cluster or cell type, we find that cell types or clusters from hypothalamus, midbrain, and parts of the hindbrain are very confined spatially to specific regions and specific small regions or nuclei, whereas cell types, clusters from pallium structures and cerebellum, they are much more widely distributed. So altogether, those characteristics suggest a dichotomy between different brain regions, with the dorsal, dorsal part, the entire dorsal part of the brain containing fewer number of cell types, but highly divergent set of cell types that are often widely distributed. And the ventral part of the brain, on the other hand, they contain numerous tiny clusters of cell types. Those cell types are spatially constrained. They are also, all those numerous clusters are also very similar to each other. So one can speculate about where this regional dichotomy come from. And one hypothesis we're entertaining right now is that maybe this is again the result of evolution. The brain regions in the ventral part of the brain, the ventral brain, has functions that are involved in the vitality, the basic basic functions of the brain that are important for the survival of the animals. And so maybe the cell types in those regions are more subject to evolutionary constraint and to less deviation, less change and less deviation. The dorsal part of the brain, on the other hand, is more involved in adaptive behavior to allow the animals to adapt to the changing environment. And for that, maybe there's then less evolutionary constraint and more reason for them to diversify rapidly during development. Then next, we want to further understand the neuronal diversity within each different brain regions, given that we observe tremendous diversity already in the entire taxonomy tree. Basically, almost 5,100 of the clusters in the out of the total 5,200 are neuronal clusters. To unravel this diversity, complexity, we then subdivided the neuronal clusters into different major structures, brain structures, and re-embedded them in UMAP and investigated their molecular relatedness together with their spatial relatedness. So in the next couple of slides, I'll show you a few examples of those sub-embeddings for different regions, and reveal again, even if you dive deeper into a particular region, you still see tremendous diversity of cell types. In this slide, it shows on the top is all the glutamatergic neuron types from the pallium, and its beautiful laminar and regional specificity. In the lower part is all the GABAergic neuron types coming from the subpallium structure, including all the cortical GABAergic interneuron types that are migrated from subpallium to pallium structures. Again, beautiful specificity here. And here in this next slide, top half shows all the neuronal types, glutamatergic and GABAergic, from hippocampus and its closely related extended amygdala areas with exquisite spatial specificity for each cell type. And the lower half shows the glutamatergic projection neuron types in the thalamus and also epithalamus, which is the lateral and medial habenular areas. And in this last one shows all the neuronal types in midbrain, hindbrain, and cerebellum. It's just extreme diversity. To further understand it, we can further subdivide it into addition and sub-embedding. For example, here we separated them into glutamatergic and GABAergic, and still there is tremendous diversity to be understood. We can also just take the midbrain out and look at the relationship between different types and neurotransmitter distribution patterns and things like that. And this is just a way for us to gain a deeper understanding of the cell type neuronal diversity. And in addition to the neuronal diversity, we also observe very interesting, beautiful diversity in non-neuronal types. And here I just use astrocytes and ependymal cells as example to show that. For astrocytes shown here, we see that astrocytes, major astrocyte populations can be divided in a major region-specific manner, from olfactory areas to telencephalon, to non-telencephalon regions, and then to cerebellum. We also observed additional unique astrocyte populations, such as the interlaminar astrocyte types that are lining the peel surface of the brain, as well as the astrocytes, a specific set of astrocytes that form the tunnel lining the rostral migratory stream for the olfactory bulb granule cells generated through adult neurogenesis to migrate from the subventricular zone from the dorsal and striatum areas all the way into the olfactory bulb itself. Similarly, for ependymal cell populations, we obtained multiple subclasses and even more clusters. Ependymal cells line all the ventricles across the entire brain. And we find beautiful correspondence between transcriptomic clusters and a spatial specificity in lining the specific ventricles in different parts of the brain. And from the data, we can also calculate the cell type composition for each brain region through an automatic computation process from our whole brain data set that has been registered to the Common Coordinate Framework, CCF. And furthermore, we can reveal the spatial distribution patterns of the individual genes, not only for those 500 genes which we have probed directly from the Murphy data, but also any of the other genes in the transcriptome through the computational imputation of the whole single-cell RNA-seq transcriptomes into the Murphy space. And here is just an example of 4 transcription factors to show their beautiful imputed brain-wide expression patterns. Finally, we find that transcription factors are really major determinants of cell type classification across the entire brain. We identify multiple transcription factor co-expression modules that are specific to major classes and subclasses and types of cells across the brain, both neuronal and non-neuronal. Okay, so with that, I would like to summarize here what I have told you about today, what we have learned from the study of a single cortical region to the entire brain. For single cortical regions, we find that cortical transcriptomic cell type taxonomies reveal hierarchical organization, coexistence of discrete and continuous variations, and similarity and difference across species. Glutamatergic neuron types are distinct between cortical areas in a two cortical area study, whereas the GABAergic neuron types are shared across areas, suggesting a developmental origin for this dichotomy. There's a parallel cell type organization between isocortex and hippocampal formation, with sets of homologous glutamatergic neuron types found in both regions, suggesting evolutionary relatedness and similar circuit organization between those two brain structures. And from here, we infer that multiple parallel neural networks can be formed by homologous sets of transcriptomic types. A whole brain transcriptomic cell type atlas reveals extreme diversity and complexity, with distinct features in different brain regions. Transcriptomic types often have regional and more fine-grained spatial specificity, revealing an integrated molecular and anatomical organization of cell types across the brain. And the relatedness between different transcriptomic types likely reflects developmental origins and or evolutionary homologies between the types. So all this can be gleaned from this even adult stage transcriptomic profiles. And we find that transcription factors are major determinants of cell type classification, even in the adult brain, in addition to their prominent roles during development, of course. So then finally, we believe that the practice, the effort of creating the mouse brain cell type atlas really sets the foundation and stage for future cross-species comparison of cell types and the establishment of a cell type atlas for the whole human brain. And we are very much looking forward to further scale up our effort to make that happen. With that, I want to thank a large team of scientists at the Allen Institute, as well as our collaborators, for making this tremendous effort happen. And the whole cell typing effort at the Institute is really a collective multidisciplinary collaboration. And I want to thank all the scientists and investigators at all different levels. With that, I want to thank you for your attention and hope you are enjoying the conference itself.